Right, so I think since we're a small group, we thought that we might start by just going around and introducing ourselves, let's say where you're from and perhaps how you're engaging with maybe your precision oncology. Fine if you're not and just interested, but to get a sense of where you're coming from and how what roles we all play in the community. So I'm Karuna Ganesh. I'm a physician scientist. I'm a medical oncologist, and I treat patients with gastrointestinal cancer for the most part. And I also run a research lab where we use single cell approaches, but also patient-derived organoid models and mouse models to study stem cell plasticity during metastasis and therapy resistance. And I'm at Memorial Sloan Kettering Cancer Center in New York, the US. Sam is co-chairing.
Yes, good morning, guys. I'm ever so sorry I can't be with you today. There's some children's stuff happening here that I need to be there for. So I'm Sam Behjati. I'm a group leader at the Wellcome Triangle Institute in Cambridge, UK. I'm also a pediatric oncologist 1 day a week. My research is broadly speaking around the origins of childhood cancer. We are a DNA RNA lab. We use single cell RNA technologies to define the differentiation state of tumor cells. And we do that in the context of trying to find a clinically meaningful signal therein, or even begin to think about how we might develop maturation treatments based on our analyses. So there are a few people in the room and a few on Zoom. Should I just go through the Zoom people here? If I shout out names and if everybody could give a one-liner, maybe just tell us who you are, where you're from, and what you do. And I think it should be manageable with the number of people, and then maybe we can go in the room. Does that sound all right? So maybe I'll just shout out names. So Sergio Aguila.
Good morning, all. I'm Sergio Aguilar. I'm from Spain, working in Holger's Gaines team, and I work at single cell level, working mostly with immune cells, and we just published this Torchir Atlas. So glad to be here.
Okay, and Yasser. Yasser. Okay. I don't, maybe can't hear us. Christoph Half. I can't see the rest of it.
Sorry, Sam. I just could unmute myself. I apologize.
Oh,
It's still 4:30 here in Montreal, and I didn't have my coffee, so I'm a bit slow with that. This is Yasser.
Good effort for dialing in.
Yeah, I don't know. I think you hear me. So I'm at McGill Genome Center. I'm a group leader here of cancer genomics at McGill Genome Center, and I'm affiliated with McGill University. I use cancer genomics. Actually, that's the focus of my research, cancer genomics, and we use a lot of single cell approaches, mainly transcriptome profiling, with a focus on renal tumors, kidney cancers, and we actually use a program, multidisciplinary program, where we look into the tumors. We develop patient-derived organoids and patient-derived xenografts, and the goal is to use these models to better understand cellular heterogeneity among cancer cells and tumor microenvironment toward finding a clinically meaningful signal, hopefully. Thank you.
Okay, Sergio, Sergio, we've got Dietmar. Dietmar, can you hear us?
I may have done it.
Mandeep.
Hi, everyone. My name is Mandeep Kaur, and I'm from University of Witwatersrand, South Africa. So single cell technologies are not really right now in mainstream in Africa, and we are still getting there. So I'm here to listen to what are the developments in the field and what is happening in HCA. My own work is focused mainly on the role of cholesterol in cancer, and I'm working on colorectal cancer, collecting samples, and I'm preparing to start a single cell project very soon in the coming year. So that is my motive to be here. Thank you.
Wonderful to meet you. Welcome. Joseph Lai. Joseph. Well, what do we move on to? Lynn? Right, Laura.
Good morning, I'm Laura Jardine. I'm from the Hanifa Lab in Newcastle. I'm a clinician scientist postdoc where I've been working on hematopoietic development using single cell RNA sequencing and then trying to use this to understand differentiation states of childhood cancer with Behjati.
Great. Mark Ahrens. Okay, I think that concludes people here. Mark is going live. Hey, Mark.
Hi, Mark Ahrens. I'm Professor of Pathology, University of Edinburgh. I'm PI for a group. We're interested in using single cell RNA sequencing for studying Crohn's disease and eventually for studying how that can progress onwards to other complications, including neoplasia.
Mark. So I think that concludes everybody in the room on Zoom. Should we move to the room, Karuna?
Yeah, so if we can all just go around very quickly, a quick one-liner, just to get a sense of now, are you clinical, are you experimental, are you computational, and how you're sort of engaging in this question. Start here.
Michael Zero, commercial genetics genomics.
I'm using Sigma in computational environments.
Karuna Chatri, experimental from Indiana University. Colorado computational from the European Public.
Michael Bonin, the science and technology advisor at Tennis.
Oh, well, yeah, I'm Moritz. I'm computational here at Vienna and Christopher.
Adele, experimental biologist at the Center.
Irene Papafeodoru, I'm computational from the European Bioinformatics Institute.
Pablo Garcia, I'm a computational biologist at HCA.
I'm Fabian Halberger from the Psychology Cancer Research Institute in Vienna. I'm also a computational biologist.
Shyam Prabhakar, the Genome Institute of Singapore, maybe 60-40 computational wet lab.
My name is Reno. I'm a computational scientist at Karuna.
My name is Amanda. I'm working in Vienna at the Spanish Research Institute. I'm an experimental person. Hi, I'm Mariana Boroni. I'm group leader of the Bioinformatics Lab at the Brazil National Cancer Institute.
Hi, I'm Luis Montano. I'm also part of the Children's Cancer Research Institute here in Vienna. And I work as a postdoc in Florian Albert.
Hi, I'm Anna Rakovi. I'm the computational PhD student from Mark Third lab here.
Hi, I'm Milan Julia, and the rest is exactly the same.
Hi, so I'm Shita, American University in Vienna, working over.
I'm Pascal Barry. I'm working from the Human Organ in Nice, France.
Ronan Chalini, I'm a director of single cell genomics at Memorial.
I'm Karen Sile, I'm a deep learning researcher at GSK, and I'm also a practicing oncologist in neuro-oncology. Fantastic. Great. So, we of course want to make the session as interactive as possible. And Simon, I thought that we might start by sort of thinking about where is the field at the moment in terms of all the atlasing efforts that are going on in the context of cancer, and also not in the context of cancer, but that are very relevant to what people are thinking about in terms of oncology. And then sort of think about from that what the opportunities people see in terms of current and future work, and how the technology is enabling that. And then also some of the challenges, right? Both technical and practical in terms of interactions and things like that, to drive clinical impact where we are now to get into that direction. And then perhaps finally think about how we can sort of, as a community, bring these interdisciplinary interactions closer together. How do we make that happen? What can the community do? What can organizations like the HCA do to encourage these interactions and move them forward in a productive direction? Am I missing anything else?
No, not at all.
Sam, do you want to get us started? Just thinking about where the field is at the moment.
Yeah, so I'll keep it reasonably brief, and it's my perspective, and I think you need to caveat my perspective with a few things. I'm not very good at keeping up with the literature, so I couldn't make any credible claim of having a complete overview of what people do around the world. That's number one to say. Number two is I'm quite interested in tumor cells. I'm not so interested in the microenvironment. So the stuff that I know is mainly about tumor cells. And with those sort of caveats, I'll just share a couple of thoughts. So I think one great thing that has happened, and I guess the other thing that I should say is I come from cancer genomics. So my PhD was at the time of the TCGA and the ICGC. So that was sort of my first exposure to international collaborations to systematically do something. And for me, the HCA is a single cell RNA sequencing version of those efforts in the context of cancer. So the first thing to say is what is really quite wonderful about the technology is that it's a bit more readily available to people. It's a bit more democratic. It's not exactly cheap, but it's not so prohibitively expensive as genome sequencing was in 2011-12. And the other thing is that the computational resources required and know-how are not that extreme as they actually still are for whole genome sequencing. So in that regard, it's been really wonderful that many people around the world have been able to use this technology, although clearly there's still an issue with exclusion of important parts of the world and their demographics. So having said that, I think what has happened is that a lot of people have used this technology to look at their favorite phenotype. I think a lot of people have used this technology to study the tumor microenvironment, which is clearly very, 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 very important. I think what hasn't happened, and this is sort of the state of, or the main issue, I think, is to use this technology and study extremely large clinical cohorts to understand clinical phenotype. And that clinical phenotype might be anything. It might be prognosis, it might be particular clinical presentations, it might be treatment response. And the one field where I think this has been done really best is the whole immune treatment sort of field, particularly checkpoint inhibition and the consequences thereof. So this is sort of how I would describe it. I would describe it as everybody has looked at their favorite tumor type in small numbers. A lot of people have done work quite extensively and beautifully on the tumor microenvironment, but the real large-scale effort on any particular tumor type, I think, is still missing, and that would be my view. So, Karuna, over to you.
Maybe I'll open it to everyone else first. I mean, what do you think? What is your experience? And does anyone want to comment on what you see as where the field is now and where there are problems, perhaps, or opportunities?
I mean, to me, the major limitation is defining the cell of origin of a tumor. Without knowing the single cell transcriptome of the normal organ, it will be hard to figure out whether the tumor-specific transcriptome is mainly because of the cell of origin of the tumor, or it is derived from the mutational signature or genome aberration. And there was a paper about 50 years ago when they compared the multi-organ, most of the tumor signature is derived from cell of origin and very little from the mutational or the genome aberration. So unless we define that, developing a targeted therapy becomes a problematic. That's mine.
Yeah, that's a great point. Others?
Yeah. So if we think, what are their real driving clinical questions? Patients dying from metastatic disease, localized disease, generally curative, surgery, or just random radiation. And so then in this context, it's this very unique challenge of a cell which shouldn't be there is able to reside in this very unusual niche. And so the real difficulty from both the scientific and experimental perspective is how do we affect the entirety of this intercommunication between two, what should be logically incompatible systems, and then we're able to crack that, we'll probably make big strides in what's needed.
So, from my standpoint, I think I view this both, having come from an experimental wet lab background, where I think the field, in terms of studying cancer biology, has historically approached the question in this way, right? You first have your favorite model system, you assume that that model captures features that are clinically relevant. And those are often big assumptions that are not actually well validated. Sometimes they are, but not very. And then you do, you perturb your favorite gene, etc. You make your discovery in your experimental model system. And then finally, the last thing before you send it out to nature is you try and find a clinical data set from somewhere to test your hypothesis. But often at that point, honestly, people are so invested in that project that it's hard to not find the answer, let's put it that way, right? Or a version of that answer. And even if, if it's not the same, if you don't find that answer, then it's easy to say, oh, there might be some interpatient heterogeneity. That's probably why I didn't find the answer, right? But that approach has, of course, had its challenges. And we know that many drugs that are discovered, if you like, based on that approach, eventually fail as they get closer to patients. And in oncology, I would argue that perhaps that is the biggest failure from a clinical standpoint, is the drug discovery pipeline is very slow, it's very painful, it's very, very expensive, and that's of course reflected in the increasing price of drugs as well, which again then becomes an equity issue. And ultimately, that's, I think, all very much interconnected to the fact that we start discovery perhaps in the wrong way. And perhaps the excitement here is that now that the field is moving towards using clinical biospecimens more extensively for primary discovery, can we move that needle, right? So can we, if you're making discoveries, generating hypotheses based on what's happening in the patient, can we then make this better, right? And to me, I think that's the excitement here. But then that then goes full circle, right? If you're making the discovery based on the patient, are we using the right samples? Do we need to have more patients, more normal tissues to have references? Absolutely. Do we need to understand normal tissue biology better? Do we need to understand the biology of metastasis better? Absolutely. And I think where the field has been is, of course, often driven by the technology. And the technology has been simplest and best for immune cells because of access and also because of dissociation protocols where single cell RNA-seq, of course, the next generation of technologies that we'll talk about will become very enabling because they don't require dissociation. They don't necessarily require fresh tissue. But given those two requirements, immune cells are the best preserved in those conditions and the easiest to study, right? And also given all the excitement about immunotherapy and oncology, as Sam was saying, I think that's where a lot of the focus has been today is on the immune microenvironment of cancers. And I think there's still a lot to be done studying the actual tumor cells themselves, and also the normal tissue cells, which often, in my disease, in intestinal cancers, the intestine doesn't like to be dissociated. It really, really hates that. And so optimizing those protocols has been challenging. And of course, very hard to also say what is a real stress signal that is biologically important and is driving whatever clinical processes going on versus what is a batch effect or what is just noise or what is garbage that should be thrown out. And I don't know that we have the answers to those questions to be able to find those signals. There's also the challenge of how expensive is this and how do we take all of these heterogeneity things and all this insight and translate that into testable hypotheses that we can then interrogate with drugs. Shyam, please.
Very nice summary. I really like your formulation of starting now with discovery from human samples. But then there's the opposite problem is, okay, what's your model system to validate?
Absolutely.
So immune cells, there are many existing model systems that those immune cells will be alive and presumably behave something like they behave in tumor, to some extent in vitro. But if your hypothesis is not about immune cells, if it's about fibroblasts, there are no good model systems, in vitro model systems. Plus, they just lose their in vivo character altogether in vitro. And so I think that's a big challenge. Start with humans, discover something, then you say all existing model systems are inadequate. And what I really need is an XYZ mouse model system. Now that will take 2 or 3 years to set up. We already got it genomics data, it can't be 2 or 3 years, right? So I think model system is a major challenge for this paradigm.
Yeah, absolutely. I think that's a really, really important point, and I completely agree. And I think the challenge is, so multiple things coming from that, right? So one is the speed, and the technology is still at, it's still early, so people can publish and people are publishing. Of course, we are all doing this, publishing data sets, not necessarily with much mechanistic validation, right? It's absolutely critical, of course, to describe what's going on. then what is the next step? And you need a model system. And what is the best model, right? And maybe that doesn't exist. Maybe that has to be invented. But is the mouse the best way to model interactions between different systems? Perhaps it is. Perhaps you need an intact animal. But then, of course, you hit this problem of what's the difference between mouse and human. Do we need better human experimental systems? A lot of us are using organoids and PDXs. We all know that those are not perfect model systems. So how do we use the single cell discovery to improve our model systems? And perhaps that should be a focus for the community. What do people think about that? Yeah.
So, starting from technology. So we have been generating over the last several years a biobank of organoids from different cancer models. In parallel, we were also generating the normal counterpart of the same region that is not tumor. So we have established the biobank of more than 1,000 organoids that are well characterized from colon, pancreas, lung, liver, so many different epithelial tissues. So this can be perhaps, this is the biobank you're using for such purposes. And I believe, this is something that we can learn.
So Fabian, perhaps we can put you on the spot a little bit. This is a fantastic resource, undoubtedly. When you say that they're characterized, I guess the question would be then, to what extent do you know what features of a tumor in vivo are preserved in vitro? And, so how do you know that? What do you do in terms of characterizing that?
So there has been a number of key publications in different organs, in different tumors like CRCs, pancreas, and CRCs from Hans Clever Lab. So we are basically a spin-off company from Hans Clever's laboratory, and the publications from his lab together with us. We have been selecting a number of representative tumors from different publications for lung, CRC, breast, to show that the organoids, to a very big extent, encapsulate the morphology, histology, mutation burden of the tumor itself. Of course, there are some models that are microsatellite stable, for example, so they develop their own mutation within the culture themselves. But most of them, as far as we could see, there is a very good representative of heterogeneity within the tumor as compared to other way organoids as compared to.
Yeah, Lisa.
I just want to ask a very naive question because I don't know a lot about this, but is there any near or far future where organoids will also include immune or vasculature or microenvironment, or is that something that will never happen?
It's a great question. I can comment on that, but anyone else want to talk about that? What are people doing or know of in the field?
There are well-established ongoing protocols that also include now bioprinting, where you can go into co-culture system implicating and mimicking somehow the tumor environment by implicating apart from organoids which represent the epithelial compartment. You bring forward fibroblasts, you bring also forward in the co-culture system immune cells. I also know some applications with microbiome, bacteria, and so on. So they are emerging now protocols. Yeah.
I can comment on that a bit more. So read our next review that's coming out soon on this topic. But so I mean, the organoids clearly are just epithelial cells at the moment, right? Which is obviously a great limiting step in the context of how we understand cancer in general. And there are two broad approaches to try and incorporate the microenvironment. One is what we might call a top-down approach, where you take tissue explants of some sort, right? You just take a chunk of tissue, and there are various approaches to do this literature, where those explants can be sort of kept alive for a limited period of time. And that can be very powerful for, if you want to test a panel of drugs, for example, or test whether your checkpoint inhibitor is going to induce an interferon gamma response. But it's much harder to do mechanistic studies to genetically manipulate these things and to observe them long term. And the other approach is the sort of reconstitution type of approach, where you can get the distinct cell components, start to put them together. But there again, it's very tricky because you don't know if you take a fibroblast out of the patient, if it still behaves the same way as a fibroblast patient, right? Or in the context of the entire host. And that, coming back to this discussion about what HCA can bring to this is, even with these sorts of reconstitution approaches, right now what we're really limited by, I think, is that we can only study the microenvironment. We can look at what's happening in that tumor, but without single cell resolution proteomics, high resolution metabolomics, we don't know what's happening systemically. We don't know what those cells are seeing and how those things are changing over time. That's a challenge. So these are things that hopefully technology will enable us to address in the future.
Within the HCA, we are also providing these organoids that are established, and their primary is more single cell. So Christopher is doing, is heading this. And within the HCA, the data that's coming from HCA organoid part, it also can be very helpful for
absolutely. That clearly needs to be done in the scale, which is fantastic for the HCA. Sam, do you want to?
I'm not really model system man, so maybe I can steer away the discussion from model systems to the question to you in the audience. Do you think you know enough about single primary tumor cell transcriptomes to think about modeling? As in, do we know what the actual thing is that we want to model? Question mark. And obviously for different diseases, there will be different questions. And then perhaps a second thing to follow on from that is, can we use clinical trials as sort of in vivo perturbation experiments?
Yasser has a question. Yeah, Yasser, did you want to? Oh, that's a question.
Actually, that was not specifically about the models, but it was more a follow-up to what Sam proposed or brought up. That is the lack of, or I don't know if we can call it a lack, but thinking of large-scale single-cell studies that I appreciate it because similar to Sam, I also worked through the TCJ and ICGC consortia, and I think I appreciate the value of large scale studies because they are very powerful, not only for discovery, but also for validation. But I think what I would like actually here, if we can discuss, is how to think. The single cell studies are quite challenging in both at methodologies of wet lab side and also data analysis, especially when we speak about cancer samples. And I don't know how we can think of finding standard or defining standard methods, defining standard approaches that allow us to run larger scale studies in different, let's say, locations that at the end, again, led us to combine data and deal with that. Perhaps that's a topic that we can discuss it somehow here. I leave it again to Sam. I appreciate why Sam brought it up, and I thought that's what I really want to discuss if we have a chance to discuss.
I think that's a really, really important point, Yasser, that you bring up, and that comes down, I guess, what lies at the heart of all the issues that we have in general. And when we say large, I think Yasser and I think of hundreds of thousands of patients rather than tens. And the issue, leaving cost aside, is of course a logistical one of getting material and processing it, and the competition of material with clinical needs, which is another whole dimension. And my gut feeling is that the only way out of it is to be able to work from fixed frozen material in either from frozen or from fresh fixed, or actually once we can work from paraffin reliably, that'll be the game changer. And I can't really see a way out of it until the technical processes are such that one can scale this up. Because so we run a couple of pipelines in hospitals where we have got a 10x in pathology departments, And despite having the 10x there and technicians there to process samples, it's just really difficult. You know, it's not a 5-minute procedure. It keeps the team busy for several hours, and therefore it sort of fundamentally is not scalable from fresh material. I wonder, could anyone in the audience maybe comment on their experience of scaling up sort of fresh tissue pipeline, or is the general view that really to do this in a big way would have to be archival?
Speaking for 10x, we have a technology solution for this. We've just introduced what we call our fixed RNA kit. And so that allows you to fix the sample at the site and then later on dissociate the single cells. But we've gone beyond that, and this is a more recent development. A lot of you might not have heard of this, but you can now take FFPE tissue blocks that have been archived, and those can be subjected to single cell analysis, so you can dissociate single cells from those blocks. And I think that's the game changer that people have been looking for. So it's not going to work on everything, of course, because some samples sit out too long before they even get fixed. But it's gonna work for a lot of samples that have previously been inaccessible.
Yeah, and following up from that, I mean, we obviously don't want to be vendor specific here, but we are precisely piloting what you've just said, because I think that will actually be a complete game changer to the whole field. And then we can even think about accessing material from international clinical trials, because it's beyond unrealistic to suggest that one could run a fresh pipeline in an international clinical trial.
Yeah, right. We're trying to align ourselves with the way clinical trials are now, which the samples get fixed and embedded. And so we've made ourselves compatible with that.
Well, the other opportunity, of course, is to go retrospectively, right? So to go back to the banked tissue archives of thousands of cases longitudinally. That's something else that we haven't touched on yet, but that I think is a rate limiting step at the moment. We can get snapshots of what's happening in patients one step at a time, but really to understand the progression of the disease, ideally you want to go back in time as well as in space, right? And FFP approaches is really going to enable that. Right?
Yes.
Yeah.
I want to make a precision, because I like very much the idea to use FFP, and probably it's also compatible with joint experiment where you will have at the same time single cell profiling and spatial profiling. There are technologies that are available for that. I would just like to mention that in my lab we have developed an approach on the isolated cell where we can combine the 10x with long read sequencing with nanopore. But with that, you can have access to the splicing events that are coming, and that's particularly important and impressive in the context of oncology. But in that case, you also need to get some samples that are compatible for this type of isolation. So I would say that we should not put all the eggs in the FFP bucket. That also consider that there are additional measurements.
Absolutely, important point.
And another technical challenge with the 10x genomics at the moment, it's not considered as type of tissue. Some tissues are very fibrotic. If there's more fibrotic tissue than your single cell studies, you are wasting a lot of time to work out. There's a lot of debris they mess up, even with the multi-ohm variability. And so I don't think a single technology will work with every tissue. So that we need to keep.
You have to try it. The FFPE changes the situation. So you just have to try it.
Do you mind if I just ask, have you done a head-to-head comparison on the same patient? Clearly, you could use tissue, like the exact same region of fresh frozen.
We don't have access to those types of samples, so we can only just buy archive samples. But I'm sure some of our customers will do that soon. This is something that's completely new.
We have done the cryopreserved single cell versus fresh single cell and overlap. They work, but also the cryopreserved, not the fresh.
Ronan, perhaps you could comment a little bit on this.
Yeah, and we try at MSK, we try different like something we believe like if you have the logic right, you get better data quality. So we try to kind of improve the logistic, but and treat the sample different way. But I feel the things you need to ask, and I join your comment is what the biological question you try to answer, and based on that, you might want to pick up the protocol that fit better your question. Karuna was mentioning that you get a lot of immune cell. That's right. If you go, for example, for the tissue that you fresh dissociate, you're gonna recover more your cells immune phenotype, because those cells survive way better dissociation. All the more fragile scientific that the cell will die. You tend to lose those ones. On the contrary, if you go to fresh snap-codon tissue and you go for nuclei extraction later, you tend to recover better all cell type, but you will lose some immune cells because immune cells are lower RNA content. So if you go for nuclei, then you get even lower number of RNA from those immune cells, and you tend to not recover those as well as cells that produce more RNA. So that's the same thing for FFP. Most of the FFP methods, whether it's spatial, whether it's a new kit from Tenex, they're all top-based, meaning that you're not capturing the whole transcriptome with the poly-PC, where when you capture with the poly-PC, it's the length RNA efficient, that's why you can do more read ONC. All the FFP methods from Tenex are top-based, so you really capture the gene you are looking at. So you have to keep that in mind, probably. Like it's probably a more scientific technique to capture some gene you are interested in. But genes that are not in the panel from Tenex, and so they claim it's full transcriptome because they try to then prop for the whole transcriptome. Maybe they are missing some gene. Maybe they are not going to recover the ribo gene the same way. So maybe won't be able to call like stress cell or dying cell the same way you are with the poly-PC capture. So I mean, everything, there is no perfect method. You just have to be mind what's your biological question, and then you have the protocol that's for the best. And in some cases, you have to deal with the sample you have at hand, and that's true. You have to clinic, most of the pipeline you get with me. So yeah, most likely if you have only 15 at hand, we know that there's some question you won't be able to answer.
Anyone from the Zoom wants to comment?
Yeah, so please, guys in the Zoom, just put up a hand in, and then Karuna, can you see the hands at your end?
Yes, I think so.
But maybe it might be easiest rather than trying both of us to moderate if you just sort of take over the moderation. Anybody else on the Zoom who would like to contribute?
Maybe I just add one comment, and perhaps maybe it's for a little bit later, but that's about the data harmonization, Sam. So we know in the I mean, you've been working with cancer cells, and I think there is no standard way how to QC data. And I don't know if we can learn anything from HCA on non-cancer tissue to perhaps develop some idea for tissue-specific data filtering that may help to translate it to cancer samples. Or yeah, that's something that I have no idea. I just brought it up to see if we can discuss this as well.
That's an interesting point. Yeah. So my view on data harmonization is this: that it's wholly unrealistic. I mean, it's certainly something that would be lovely to have in place, that everybody agrees on a common set of standards. The other thing that I would really love beyond data harmonization is that we have minimum quality criteria below which we do not publish. We have downloaded data from other people, and the gene count per cell has been so atrocious that certainly in my group I wouldn't let anybody work with that data, let alone publish it. So that's a huge issue. And let's not even go to the gene fraction, but just on the very basic unit of gene count per cell, it's some of the stuff out there is pretty atrocious. So we haven't even reached the point yet where the data is consistently of good quality, and then applying QC filters as a standard would be a wonderful aspiration, but I think it's wholly unrealistic. And the issue is this, and I think this is why people, why some data that is out there is not ideal. It's just bloody difficult to get really good data. And that's just the reality of it. It's just very difficult to work with it, with specimens. And it would be lovely if publishers at least work towards a thing to say, you know, unless your data meets these criteria, we wouldn't consider that to be publishable data. It's a bit like, you know, when you submit a Western blot and it's smeared all over the place, no one would publish that because everybody would agree that it's not a publishable Western blot. This is not data that we should entertain to be adequate, but somehow, because in single-cell RNA sequencing world, it's not a wishy-washy thing, it gets through. In cancer genomics, it didn't get through, I think, because rubbish data presents as basically a lot of artifactual mutations. So it's quite easy to pick out the rubbish samples, but somehow in single-cell RNA sequencing that hasn't quite got through, and it's a huge concern. Maybe anybody else, any thoughts on QC, harmonization, that sort of thing?
Sam, thanks for bringing up that topic. It's kind of a something we've noticed as well quite a bit. There are lots of published cancer single cell data sets where some cell type, they essentially, just like you, I would say, throw this out, this bad data. Don't even try to draw any conclusions from it. The problem with single cell is it's so expensive, not like a Western blot where you can do it again. Like, okay, you spent all your money, and somehow get a paper out of it. That's the psychology. But really, what we've seen in our analysis is that if you do stricter QC, so you may lose half your cell, okay? And then the postdoc who generated that data set is in tears, but your conclusions are much better. And it sounds paradoxical maybe, but if you throw away half your cells at a bad quality, the remaining half your cells give you much crisper, more believable conclusions. And so
I couldn't agree more with you. What I say to my team, you know, you're better off with 100 high quality cells than you know 2,000 absolute garbage, rubbish, blobby things in the middle. You know, no, you're absolutely right. It's just a shame that that number imperative seems to sometimes override these concerns.
Okay, can I ask just as a counter to that? So we study metastasis, which is obviously, you know, the cells are changing not because they want to, but because they're stressed and they have no choice, right? And so how do you distinguish between real biologically meaningful stress versus garbage data? And I don't know, how does the field deal with this? Because we grapple with this all the time. I'm not sure we have a good answer. Ron.
I would say you go for nuclei.
I think
not for the nuclei preparation, because then you know that by start freezing your tissue, you just preserve the tissue as it is at the moment you do the resection. And you're not kind of doing 1 hour, 37-degree incubation that will stress all the cell, and then you're hiding your real biological stress signal with the stress signal you're inducing by the same thing. So again, if your question is like, I want to see what cell are stressed in my tissue, then this defines which method you want.
Great. The fundamental challenge for this from the analysis perspective is we've got no way of distinguishing biological versus technical skill, and there's no statistical methods. The one thing is that perhaps after 5 years or so with HCA data, when we have a better idea of what is the distribution of expression in a single cell, we may be more reliably able to identify outliers, but then still we'll have that challenge of distinguishing biological outlier versus the will be versus the technical. So I think it's just going to be a iterative process.
Yeah. Lisa.
So what if you see this, for example, the stress effect consistently across 10 metastases, and you don't see it in whatever control samples, would you still think this is a possibility of pure batch effect, or would that basically tell you this is real biology?
I think, you know, at the moment, honestly, the way we interpret it is still based on our biological knowledge, right? Yeah. Think about like, what do we already know about the biology of the cells from other systems? And so does this make sense? But that's perhaps not the most elegant way to do it, right? But maybe that's our answer.
But reproducibility is not enough in itself. Yeah. Just seeing the same pattern across samples.
But if you process all your tissue the same way, maybe the same tissue processing artifacts can apply to all your samples, right? Chance.
I think stress is a very interesting question. Hard to say is it biological or is it your processing, but perhaps what Sam was alluding to and what I was alluding to was something much more basic. Are you detecting 4,000 genes in your cancer cell? Are you detecting 400 genes in your cancer cell? And it's 400, 500, you should probably throw it out and keep the cells that have 4,000 genes, something like that. And we found that that just has a profound impact on our ability to see patient-to-patient variability in other patterns.
Exactly. Since we're on data processing and analysis, I guess perhaps the question that comes up for us is, one is about batch effects, and love to hear more about what the computational people in the world in the room think about that, because when we're dealing with real world tumors, right, each patient has their own set of mutations, as well as all of the other clinical variables, all the patient variables, what they ate for breakfast, all of that stuff, right? So how do you deal with correction, integration, batch in this context?
Yeah, well, I can comment, but again, I think I'm thinking from the Atlas perspective, where you have many different data sets that might look at the same thing, but have protocols, samples, and so there, I would repeat what I just said, which is I would think if you see a pattern consistently across data sets that have very different protocols that they use, that for me would be the most solid way to distinguish batch from wild. As soon as you have very few samples, that becomes a big problem, and you have to go back to very long. Yeah.
I think another way would be much more expensive is to follow it up with the spatial transcriptomics or spatial proteomics of your selected sample the probes. So that way you can take care of the batch effect, the stress effect, if you have to process the sample in the right way. So probably that would solve the problem. But again, is it practical to do both single cell first and then develop your probe set and go back and analyze the same sample with the spatial technology? I don't know how practical it will be money-wise.
Yeah. Maybe I can comment a little bit on that, because we recently were able to integrate at least 13 studies of tumor from different labs, also from different technologies. And in our case, to efficiently remove the batch effect, we had to consider each patient and each technology as a batch. So in the end, we were able to integrate in a nice way the immune cells, but I think we have lost several important biological parameters from each patient. So yeah, it's not always a win-win. So in our case, we were more interested in the immune cells, so it worked pretty well. But I think if we really want to characterize different biological specifics of patients, that is not the right way.
Thinking from the computational perspective, data batch correction is the harmony algorithm that's used the most, and it's fundamentally this expectation maximization, which is already commonly used in statistical genomics. The challenge is it's very computational and slow. So as we're trying to scale up, that's going to become very difficult. I think one of the challenges is that we're trying to integrate it with other modalities, just like spatial transcriptomics. Then we face even a greater challenge of an even more like zero inflated distribution. So if anything, we want a technology where we're going away from that problem.
Oh, Sam, go ahead.
Oh, thank you. I was going to say my thoughts on batch correction, and for what it's worth, are these: that I think sometimes we may be getting a little bit over obsessed with having ourselves sort of form a common island on a UMAP. In my head, cancer cells from different patients that needn't necessarily biologically be sufficiently similar so that even if there was no batch, they would integrate. For me, the test has always been this. So if I look at the non-cancer cells and immune cells from different patients, form of the same type. So let's say CD8 cells and macrophages from different patients mixed together, then I know that fundamentally, whatever the batch is probably not so strong as to artificially separate cancer cells, and therefore it might not be unreasonable to suggest that a separation of patient cancer cells is biologically driven, because there is a control. There's always this inherent control of non-tumor cells, which should mix itself the same type. That's sort of my view on it. And I feel that any kind of batch correction has a danger of then distorting all subsequent data analyses. And I'm perfectly happy with having cancer cells from different patients segregate, provided that the non-tumor cells mix nicely together. That's one thing to say is, and the other thing is that in our experience, cancer cells from different patients that do mix are very rarely some, wonderfully rare stem cells or whatever. They tend to be the rubbish cells within each tumor samples that tend to congregate in the middle of a UMAP. That's kind of my 2 pence on it, but you may entirely disagree.
Again, very similar point to Sam. The main point to note is it's trial and error. There's no one batch correction formula you can use for all cell types, all tumor types, all cohorts. And then even if you use the same strategy, different algorithms can give you dramatically different results, qualitative behavior. So just trial and error. From a lot of trial and error, what we've seen is that if you want to look at patient-to-patient differences, which is a key thing we want to learn, treating each patient as a batch really smashes, collapses those differences. You lose the signal. So at least for cancer cells, when you treated each patient as a batch, all we were left with was the cell cycle variation. There's nothing else interpretable in our data, in our cancer cell data at that point, but the immune cell data looked quite nice, as you were saying. So really kind of cell type. Also want to maybe give my version of what Sam was saying, which is that the whole batch effect correction, the whole thing started with samples where you know that the biology is the same, and that's more or less the same. And that paradigm may not be even a valid paradigm where each patient is different. So what we've done is a kind of fallback position that's in between. To say, don't say each patient is a batch, don't ignore batch altogether. Say that each cohort is a batch, for example. And then we try to subtract out cohort-specific effects rather than patient-specific effects. And that we found was good enough.
Great, that's very helpful. It's actually, I have to say, as an oncologist, it's very enthusing to see the single cell community sort of coming to this perspective, right? Because I think in the early days, when we were talking to computational biologists, they were like, oh, but this is all batch effect, this is all different. Yes, they're cancer patients, of course they're going to be different, but they're supposed to be different, right? But it's nice to see the worlds coming together. It's actually a very exciting thing, I think people are understanding the interdisciplinary questions better, I think, over time, as the fields are all sort of working together more, right? Which I think is very exciting. But perhaps, yes, go on, please.
Sorry, I want to slightly move to the target discovery.
Great.
Yeah. So one of the things as a pharma company we are really interested in is synthetic lethality concept. Yeah. And is anyone, this might sound crazy, but is anyone already thinking or using single cell data sets of tumors that certain cell proportions indicate certain vulnerabilities or certain immune correlates indicate certain vulnerabilities in these tumors using these single cell data set? Is it a good question in first place to ask using this data set? Perhaps combining the single cell with the dependency maps that are available from large scale CRISPR map-out studies. Yeah, it's just a question that I've been thinking a lot, but we never got the chance to really explore this further. But I was wondering if anyone is thinking in this direction.
That's a great question. Comment?
I think a lot of people who are working on HR deficiency are the one. That's where the synthetic lethality drug combinations are effective in clinic. That's one most effective way only the HR deficiency, in my knowledge.
Which division?
The homologous recombination score.
I think you're raising a number of broader questions, I think, which are interrelated. So I think one is the question of how do we move from single cell atlases to target discovery? And that's an important question that would be great to address. I don't know, would people like to comment on that? I mean, once you have your atlas and you've decided how to QC it and define what's going on, how do you move from that? Targets are of course important, but even from the atlas to testable hypothesis, how do you do that? How do everyone approach that? Sam, how do you approach it?
To be quite honest with you, Karuna, I haven't got any particular thoughts on it. So I was hoping that someone from the audience will provide a more informed answer than I would.
Maybe I can comment on how we do it. Right? Or, we start, I guess, in the literature, right? I think, again, because the field is very much in its infancy, often people describe their favorite system. And then I think, honestly, I think this is a challenge for the field is developing perhaps algorithms to help us identify, of these trajectories that we're seeing. And I think there are good algorithms in some cases to describe trajectories. But then it's hard to get from that to say, like, how do we find the critical node that we can perturb, right? Or that we should perturb. And that's sort of coming to the point that you're making. I mean, with synthetic lethality, if you look at the depth map or the Sanger equivalent of it, people have done genetic experiments to define changes in other things, right? Dependencies. But then how does that translate to the transcriptome space? I don't think we really know yet, right? And those experiments perhaps need to be done at some point. And is the transcriptome the right readout for those experiments? We don't know. But then this then comes to the perturbation experiments, which I think are just starting to be done in the field at scale. And we've heard some of that this morning, right? And there again, I would argue that we need to write models to do those perturbations. And is the depth map the right way to do those experiments? I don't know, right? And in some cases, the answer is probably yes, and in others, probably no, right? So I think maybe we can step back and think just about trajectories. So a lot of these trajectory analysis techniques have been developed on normal tissue atlases, right, appropriately, where there's already a lot of prior knowledge about development of trajectories, especially in the immune system, but lung systems as well. To what extent do those trajectories apply in cancer, where perhaps those normal trajectories have been perturbed? What do people think about that?
Maybe I will say something, Karuna. So I think it's going to be a really long-winded answer. So apologies at the outset that I'm going to waffle now for about 5 minutes. The issue with trajectory analysis, in my view, is as follows. So at the end of the day, if you think about normal tissue, we have some sort of pre-existing knowledge which allows us to ground our trajectory analysis in something. So if you think about the kidney, we know reasonably well that that these are the early cell types, these are the late cell types. In normal hematopoiesis, there's a very clear hierarchy. So one can use that pre-existing knowledge to then sort out the transcriptional detail of the trajectory. To derive a de novo trajectory on something that we don't know in normal tissue, I don't actually think it's technically possible, because everything that we do, pseudotime or velocity, all of that, although obviously velocity is biologically more informed, but all those methods fundamentally are not a true biological longitudinal readout, because the tissue that you have is a cross section. So that's sort of one caveat around it. Now, if we now think about cancer, I don't really think we have a clue what the trajectory is. So let's say the notion of de-differentiation in adult epithelial cancers, we've just done a huge analysis across the whole TCGA data on this in bulk using single cell data. That notion of de-differentiation is very difficult to uphold in transcriptional terms. If the idea is it de-differentiates to something fetal, it just isn't true. In the context, so the problem here then is we don't actually know, we haven't got anything to anchor the thing in. And I think that's a huge issue. Or the idea of a cancer stem cell, that may be true for some cancer types, but it certainly is not universally proven. And also the identity of those is not known. And that makes then drawing a trajectory from cross-sectional data very difficult. Now, in pediatrics, it's ever so slightly different, because we do know in pediatrics that childhood tumors do recapitulate human development, and therefore we can use the human, what we know from the human development cell atlas, we can use those normal trajectories as an anchor point and something to project the cell types onto. So I think that works quite well, but I'm highly suspicious about other contexts, simply because we haven't got enough pre-existing knowledge to anchor our trajectory analysis in. Maybe if one did longitudinal sampling in the same patient, so you've got the primary and then the met and then another met and then it regrows and that sort of thing, then we can begin to get actual real longitudinal time points and then try to ask the question, does anything necessarily evolve? But even that is a huge assumption, to say that a tumor evolves. A tumor doesn't necessarily evolve. Of course, it acquires additional somatic mutations and it changes. It goes from primary to metastatic site, but that needn't necessarily be a biological evolution. It can be a stochastic event of a single cell detaching, going somewhere and finding a nest. So actually, I think the trajectory stuff on cancer, particularly adult cancer, is totally difficult to discuss.
So is this more about trajectories that are common across patients, or trajectories within one patient from the healthy to whatever cancer cell you encounter once you cover some? Or
did you hear that?
I think it's both. I think it's intra-patient to understand how things change within a patient, but actually, I think what most of us are interested in is this sort of holy grail of there's a fundamental canonical trajectory that one might identify in the development of each cancer.
because there are some cancer types, I thought like colorectal cancer, where there is at least genetically some sort of common paths to.
Yeah, so you're quite right. So that then goes back to the modeling. So here you could see a beautiful way where you could say, well, I'm going to take whatever model I have, and I'm going to introduce each mutation successively, eventually transform the cell, and then get cross-sectional readouts at every point of somatic evolution, for lack of a better word. So I think that's absolutely something doable. I'm not quite clear how we do that from patients. So if you look for colorectal cancer, maybe within a patient or maybe within a tumor, where you get more polyglot components. Components, you could sort of go there.
Yeah. So that again, I think comes back to this idea of needing longitudinal samples, right? So in many cancers, it is possible, in principle, to design studies where you get pre-cancers, and then eventually when those patients go on to develop real cancer, then to get those and then see how those things may have evolved, or at least been selected for. And similar things, of course, the easiest studies, if you like, the lowest hanging fruit, which is starting to happen now, we heard about that from Eli earlier today, are the perturbations pre and post treatment, or pre-on and post treatment with certain drugs. And there, of course, in the clinical oncology space, that requires strong collaborations with pharma companies who are often sponsoring these trials to access those samples, and a lot of this analysis is happening in pharma companies, of course, to understand what's happening, at least with that single perturbation of the drug. But then that has to kind of come back to how we think about analyses. But I'll say, I mean, the point of colon cancer, right? But we know that there's a stepwise series of mutations. It's not always the case, but it's not just mutations, right? There's a lot of stuff that is not mutational that is happening, and we know certainly in most cancers, as you transition from early stage cancers to late stage cancers, there's not really a lot of acquisition of new mutations, but the transcriptome is evolving, and we don't really understand the mechanisms very well why. And then it then gets harder to model that, right? Because we don't know exactly what those stresses were that induced the change. And so then again, how do you make assumptions about trajectories? And I would say in our colon cancer data sets, we are unable to use any of the existing trajectory algorithms to accurately map the structure.
Yeah, I know that there are also some algorithms being developed, and I think also already published that do this trajectory modeling on the patient level, where you don't necessarily need longitudinal sampling, assuming that there's a common trajectory between patients, of course. But then you can combine information between patients to try to sort of align them along the trajectory. Absolutely. You would need a lot of sampling.
Yeah. Many comments.
Hi, I just wanted to make a comment regarding this longitudinal sampling. I think it's great to try to get a clinical trial information first, but there's a lot of governments already where they are collecting data in the national health system.
Absolutely.
In which they just sample patients, and they have like volunteers that come and donate blood once a week, once a month or something. And then some of these patients, some of them will have cancer or have other diseases. So you can get now backtracks to all the samples that they had before they had the cancer and afterwards. And these are incredibly rich data sets. So some of the governments, which are doing this very well, for example, the government of Denmark, they do it super well. And that could be super helpful to get insights about heterogeneity.
Absolutely, these are phenomenal resources. But the UK Biobank has been doing a lot of exciting things, and Singapore has an effort and so on. There are a couple of points.
I just want to clarify, probably this is specifically the pseudo-time inference that we're discussing, or just more general?
I think more generally, just thinking about how do cells get from one stage to another, right? And while there are smooth trajectories in those state transitions in development, they don't necessarily apply in cancer.
I mean, to get the best data, you need to do the multi-region sampling, which is not practical.
Good point.
Right.
Yeah.
So even if you do the analysis pre and post treatment, it's not guaranteed that you are capturing every information. That limitation we need to keep in mind when we develop the models.
Absolutely, and perhaps there the only way to get good data is from rapid autopsy programs, right?
Exactly.
And there it has its own set of limitations in terms of how long is it sitting, sitting around. Okay, so I think we have a few minutes. So Sam, do you want to see how we want to end this?
I was just wondering, is anybody doing something really very large scale? I'd be really sort of enthusiastic to hear that. Or are we all in the 10, 20, 30, 40, 50 kind of end scenario?
What do you think the scale would be that you'd like to see?
I don't know. It's obviously highly tumor type dependent. Some of the tumors that I study, there are 2 or 3 patients per year in the United Kingdom. But I was wondering, is there someone out there doing 500 breast cancers? Is there? Or 500 colorectal cancers, or some common epithelial adult cancer at scale, and perhaps even within the context of a trial? I take the silence as a no. Yeah, Yasser, you're about to say something. I'm excited to.
I just turned it on. No, it's not exactly single cell, but in the context of a spatial transcriptome profiling, we are kind of working toward that scale, but it's not yet single cell resolution, as you can imagine.
So is this Visio 1 FFPE? Is that what you're aiming for?
Exactly, that's the FFPE.
Okay.
That's great. So perhaps great to hear your thoughts on what do you see are the biggest opportunities? What with the new technologies that are coming out, with the opportunities to perturb, etc., that are coming out, what do you see are the biggest opportunities for single cell genomics and precision oncology? How do you see it?
And start to comment on that. I feel so far with single cell RNA-seq, the red technology you can go through hospital and single cell level. The main limitation for me to apply that clinic is the number of cells you capture, for example, like you really see the tip of the iceberg. So if you think in the context of resistance, therapy and everything, you expect fewer clones that you resist treatment. I mean, you get only 10 or 20,000 cells in a sample, you might miss those cells. If you think in terms of in situ distillation, you're going to capture less genes, so it's not full transcriptome, but now you can capture millions of cells in one slide. And you probably will be able to see better the full spectrum of one cancer in one patient. So I think these are the big kind of opportunity for future. And also, if you tell in terms of logistics and pipeline, I'm a technical person, so I always think about logistics. This is what already all the biologists are doing before, right? They take the tumors, they fix it, they embed it in FFPE, and then they do some staining, or they do HNE, and they do a diagnostic, and they can kind of tell the patient what type of cancer they have. So now if you do that in the context of in situization, it's exactly the bivalent standard. So from in situization is really the future. It's something that can directly almost be applied in the contrary to single cell RNA-seq, which has been challenging.
Yeah, that's great. Others? Anyone on the Zoom?
From my point of view, the only thing that I would contribute, and I think we said it in one way or the other, is the greatest opportunity lies in going to archival material from clinical trials and to marry up RNA signals with whatever clinical parameter the trial measured, because it would seem highly likely that the cancer cell state or the microenvironment state encodes for something clinically useful. One just needs to think carefully about the tumor type, the trial, and the question. But this is where the power, in my head, of archival material lies.
If I may add the comment, yes, I think we have to continue your comment more to pathologists and surgeons, because a lot of times for research that is left is the leftover from the pathology, and a lot of times we cannot even access the material because it has to be kept in the hospital diagnostic. So perhaps we have to revisit again how sample collection is done. Like if we do needle biopsy, especially we want to capture the data, there is immunity, etc. Questions, you just get the leftover pathology. It's not always, it's good. I'm always happy to get the leftover pathology, but we need to rethink again how we organize the hospital. And I think if I from hospital, I think perhaps we have to make it some time.
I guess I'll say that, I think all of these things, many of these things are happening. The technology, the samples are going to get there, etc. The paraffin stuff. I guess for me, the bigger opportunity / challenge is how do we go from all these data that we're going to have to clinical impact, right? And so what are our questions? We want to identify biomarkers. Do we want to identify targets for drug discovery? So how do we move from the description side to the action side? And for that, I think we kind of all need to work together, right? I think the challenge for our computational colleagues is really how do we get to testable hypotheses from these data? And are there ways in which machine learning can help us to do that and to prioritize targets? Because yes, you can have 500 genes change, but you can't test those 500 targets. So how do you actually do that in a meaningful, actionable way? And then how do you then get that back to patients? And the other related issue is cost, right? How can we bring down the cost of these technologies such that it's actually accessible and meaningful in a real world context for everybody?
I think when we get to the 500 scale or something like that, let's say TCGA scale, then we can start correlating with outcomes. And then but with single cell data and large data, then I think we'll have a much better set of candidates to say, we know this gene is important or highly likely to be relevant. But right now, the descriptive phase, all we can say is we have cell types.
Yeah.
And we have patient types. We can at least learn patient types. Still without learning how to treat them.
Yeah.
But that's still, then we can stratify retrospectively. What we're trying to do is retrospectively go to clinical trials and identify the patient types that were not known before and then say, do those patient types respond differently? That could at least lead to some concrete repurposing or more tailored prescription.
That's great. And then maybe you don't need to do the whole genome. You can do a targeted fish for 20 genes or whatever it is on those retrospective samples.
Exactly.
Absolutely. That's great. Any other final thoughts?
From the computational side, are we still relying on the things like ingenuity pathway for the identifying signaling node in a cell type, or are there anything else you're building computational side to make it more relevant clinically?
So relying on what?
At the moment, we all go through the ingenuity pathway to figure out what is the signaling node coming from this differentially expressed gene in the cell line. Do we have anything better than that? For the, it can be clinically applicable to find out a signaling node that the cell is dependent on.
So one of the things we usually do, other than relying on just identifying pathway, is quantifying certain gene signatures. If you have a transcriptome, or even if you don't have a transcriptome, probably you can just measure the expression of your 20 genes that we actually predefine for a phenotype, and then see how this score is changing for the patient. And if based on certain cutoffs, of course, the previously defined cutoff, that where exactly the signature is giving kind of survival impact, and then probably using that cutoff for these 20 genes as a combined score, not individual score, combined score, and then prioritizing certain patient populations. So it's completely, the pathway is good. It makes a good figure, and you can see fancy things there. But I mean, if you go into the actual clinic, quantifying pathway will not be as effective as quantifying gene signature itself. So that's what we do, and this is kind of proxy for whole oncogenic profile of the patient.
Fantastic. Sam, any final words?
No, not at all. Just thank you everybody for participating.
Thank you everyone that was a great discussion, and hopefully look forward to working with all of you in the community. Let's hopefully make an impact on patients someday. Thank you.
Thank you.
Karuna, should we chat briefly once everybody has left?
All right, we have two wonderful